All right, we're trying something slightly different. I'm recording in class, so if you hear background noise, it's not my cats this time, it's students. So we're going to talk about the cell cycle, which is basically mitosis, as you see in this meme here. So before we get into mitosis, we need to talk about the cell cycle. What is the cell cycle? It is basically the life cycle of a cell. It's a highly ordered, highly structured series of events. It happens in eukaryotic cells, and it is very different from the life cycle of bacteria and certain other cells, but we're not going to talk about those. So it consists of interphase, which is where a cell is going to spend most of its lifespan. Interphase consists of G1, S, and G2, which we'll talk about in a moment. Then there is mitosis, followed by cytokinesis. You see a nice diagram of the cell cycle here, including something that we'll talk about, which is G0. So let's talk about G1 phase. Technically, the G stands for gap. I also tend to tell my freshmen to think of it as standing for growth, because a lot of growth of cells does happen during this time. Notice that the cell that I have pictured here is growing as we pass through G1. So the cell is going to perform whatever functions it normally performs. It's metabolically active, and that is a point at which the cell is going to also grow to its normal size. So it's going to be duplicating organelles, it's going to be making more proteins, etc., the things that it needs in order to be functional. After G1 phase, you have S phase. The S in S phase stands for synthesis, which is when DNA is synthesized or replicated. So DNA is going to be in the form of chromatin during all of interphase. It's going to be stringy. It's not going to be condensed. And then you're going to have DNA replication happen during S phase, at which point you're going to produce these two long uncondensed chromosomes that are still going to be connected at a centromere. So later on in the cell cycle, when they condense, you'll see the centromere actually show up, which we'll talk about in a moment. After S phase, you're going to have a pair of sister chromatids, which are two identical replicated copies of a chromosome. They are attached at the centromere, which in eukaryotic chromosomes is this kind of narrow area on condensed chromosomes, but it's where sister chromatids attach to each other, and it's also going to be where spindle fibers attach during mitosis. Let's talk about the centromere for a second and what's going to happen later in mitosis. You're going to have this structure formed at the centromere called the kinetochore. The kinetochore is a complex of proteins that are going to bind to the centromere, and they're also going to bind to microtubules, which are these proteins that make up the spindle fibers. When the microtubules are bound to the kinetochore, that's going to allow the spindle fibers to pull the kinetochores and pull centromeres of these duplicated chromosomes apart. So this is where the actual movement's going to happen during mitosis. And the reason why I go over this is there have been one or two free response questions on the AP test where they talk about kinetochores. After S phase, you're going to have G2 phase, which is the gap2 phase. This is when a cell, before S phase, a cell has already committed to replicating. So G2 is going to be primarily preparation for actual cell division itself. This is where you're going to have a lot of protein synthesis happen to produce the proteins that are going to be necessary. You're going to produce a lot of ATP to power mitosis, and you're also going to replicate centrosomes. Centrosomes are structures that are composed of, in animal cells at least, a centriole, which are, I say they kind of look like churros, but they are these structures that are found in animal cells. In non-animal cells, you'll also find centrosomes, which will have what are called microtubule organizing centers, or the MTOC. And that's what's going to form the spindle fibers that are going to pull the chromosomes and pull the chromatids apart during mitosis. You're also going to have extra cytoplasm, extra organelles produced in preparation for mitosis. But if a cell is not going to undergo mitosis, it may choose to exit the cell cycle, or it can also pause at various points in the cell cycle. And in our next lesson, we're going to talk about checkpoints, which would be another location to pause the cell cycle. Cells can enter a stage during G1 called G0, which is where they are non-dividing. And this can be either reversible or irreversible, depending on the specific cell type. So depending on external clues like growth factors or other cell signals, some cells can exit G0 and re-enter the cell cycle. These are things like stem cells, liver cells, which can reproduce. Other cells will permanently exit the cell cycle, and the rest of the lifespan of a human, they will not divide again. So, neurons, skeletal muscle, and cardiac muscle cells, once they're mature, they do not divide again. But cells that are dividing are going to exit G2 and end up going through mitosis. Mitosis is technically nuclear division. It is dividing up the chromosomes in a cell to make sure that each cell that is produced is going to be genetically identical to the original and is going to have a complete copy of the genome with every single chromosome found in the original cell. This stage is going to alternate with interphase in the cell cycle. So mitosis alternates with interphase. So if you look at the cell cycle, you could think of it as this alternating sequence of interphase, mitosis, interphase, mitosis. Although I will say that cells do spend the majority of their lifespan, the majority of that time is spent in interphase. And mitosis is just a brief blip in the life cycle of a cell. We just spend a lot of time talking about it because there are a lot of events happening during mitosis. Mitosis is going to allow for reproduction of cells in a multicellular organism. That's going to be growth, tissue repair, anything you need new cells for. And if you have a unicellular eukaryote, that's going to be responsible for asexual reproduction. There are 4 phases during mitosis: prophase, metaphase, anaphase, and telophase. And then it's going to be followed by cytokinesis. So during prophase, a number of things happen. First, what's going to happen is the sister chromatids are going to condense, and you're going to actually see those X-shaped chromosomes that we're used to thinking about. The nuclear membrane disappears, so the nuclear envelope goes away. That way there's enough room to separate these chromosomes, and you can separate them into different cells without the nuclear envelope getting in the way. The mitotic, also known as the protein spindle, begins to form between the centrioles. The centrioles and centrosomes move to opposite ends of the cell. And then next is metaphase. So I say prophase stands for preparation. You are preparing for the rest of mitosis. Metaphase, you're going to have your chromosomes line up in the middle of the cell. If you want to use technical terms, the chromosomes are going to attach to the spindle fibers at their centromeres. Then they're going to line up along the equator or the middle of the cell. Anaphase is when these chromosomes are pulled apart from each other or they move away from each other. So notice the cell also begins to elongate at this point. It's getting larger in preparation for division. And you have your chromatids moving apart from each other. So they are pulled by their centromeres, pulled by the kinetochores that are attached to the centromeres, and the spindle fibers are going to pull those chromatids away from each other towards opposite ends of the cell. At telophase, you have the mitotic spindle beginning to break down. It's no longer necessary. You're going to have nuclear envelopes start to develop around your chromosomes, and then you're going to have the cytoplasm start to divide. Telophase, in some cases, we tend to say stands for 2 nuclei. You're going to see 2 nuclei forming at that point. I am going to let you know that telophase is the last part of mitosis, and it overlaps with the next part of the cell cycle, which is cytokinesis. So you'll start to see the cell actually dividing the cytoplasm up while telophase is happening. And for some reason it didn't show up, but there is a second nuclear membrane forming around this second set of chromosomes. Cytokinesis is just the splitting apart of the cytoplasm. It is not technically part of mitosis. Mitosis is nuclear division. Cytokinesis is cytoplasmic division. So cytoplasmic division is going to complete the process of cell division, and it's going to happen differently in different types of cells. In animal cells, you're going to have something called the cleavage furrow form. It is a band of microtubule proteins that are going to constrict in between the 2 forming nuclei. They are going to pull, I think of it almost like if you took a particularly heavy set person and you put a belt around them and you just kept tightening it until they squeezed apart. It's a very morbid example, but that's kind of like what the cleavage furrow does. Then, in plant cells, since they don't have as flexible an outer surface since they have that cell wall. You're going to instead form something called a cell plate. So these little vesicles start to gather in between the two sets of chromosomes. They join together and they form this cell plate, which will then end up forming into the cell membranes that are going to separate the cell, and a cell wall is going to form in between them as well to produce these two new plant cells. All right, that's it. I hope you enjoy the grotesquely dividing face on here, and we'll have our next lesson on regulating the cell cycle soon.